this is going to be our video intro or video for cellular respiration the whole way through. So cellular respiration, we're going to make ATP by breaking down sugars. So remember that when we think of ATP, we are thinking of energy. And then cellular respiration is aerobic and it's going to require oxygen, while the other definition we're going to look at later is anaerobic, and that's without oxygen. And aerobic stages are going to take place in the mitochondria of either animals or plant cells. Now, cellular respiration occurs in three reactions. We have glycolysis, citric acid cycle, and the electron transport chain. You should be able to know what goes in, what leaves, and where each of those molecules go throughout. So our overall process, or overall equation of this process, is C6H12O6, which is glucose, plus 6O2, which is oxygen, is going to yield or make 6CO2 and 6H2O. The CO2 is carbon dioxide and H2O is water. The reactants are on the left, while the products are here on the right. And as we see in photosynthesis, cellular respiration, they are inversely related. The reactants of cellular respiration are the products of photosynthesis, and vice versa. So our first stage of all of this is glycolysis, and it's needed in cellular respiration. We cannot do anything in cellular respiration or in fermentation without glycolysis. And glucose is broken down into the cytoplasm to produce ATP and NADH. So we have a 6-carbon glucose here. We're going to put in 2 ATP, or energy required, to break it down into 2 3-carbon molecules. This is an intermediate. Then we're going to make 4 ATP from this, and also 2 NADH. Remember, if I see H or hydrogen, I'm thinking electron carrier. So NAD plus is electron carrier, NADH is after I've added the electron to the electron carrier to come up with our 2-pyruvate. So here's a sample question that we looked at in class. And here we go. So this first part here is glycolysis. We've already talked about that. Then we have 2 different ways we can go. We can go with oxygen, or aerobic, or no oxygen, anaerobic, aka fermentation. And that's what we're going to talk about here next. So aerobic respiration takes place in 2 stages. We have the Krebs cycle and the citric acid cycle. That's the same thing. And then we have the electron transport chain. We have aerobic respiration in prokaryotes, and that takes place in the cytosol. Aerobic respiration in eukaryotes take place in the mitochondria. Now, most of the time, we're going to think of aerobic respiration in eukaryotes. And then here's another example question for you. Kind of going back through what we just talked about. So fermentation allows glycolysis to continue while making ATP and oxygen is unavailable. The function of fermentation is to recycle NAD plus from NAD from NADH. This is what can keep the cycle going back and forth. Fermentation is an anaerobic process, and it occurs when oxygen is not available for cellular respiration. It does not produce ATP. So this is the only thing we're doing this for, is to recycle NAD plus from NADH. So our first one is found in animals, specifically mussels, and it's called lactic acid fermentation. We're still going to have glycolysis. We're still going to make 2-pyruvate. We're still going to have making 2 ATP and 2 NADH in glycolysis. The only thing is we're now adding the hydrogens into the pyruvate to make lactic acid, and the NAD plus is going to go back into glycolysis to be reused, and this is both going to occur in the cytoplasm of mussels. So alcoholic fermentation is similar to lactic acid, just going to occur in different organisms, and glycolysis splits into glucose, or splits glucose into 2-pyruvate like we did last time. This time, instead of making lactic acid, we're making 2 carbon molecules called alcohol and 2 carbon dioxides. NAD plus is going to recycle back in the cytoplasm, so this cycle can keep going. And here's an example for you as well again. So fermentation can be used in food products like yogurt, bread, and cheese. So in eukaryotic cells, Okay, we have enough oxygen, we have mitochondria, so we're going to transport this pyruvate into the mitochondrial matrix using 2-ATP, or what we call active transport. Now, citric acid cycle or Krebs cycle is going to take this 2-pyruvate and they're going to break them down into carbon dioxide and energy-carrying molecules known as NADH and FADH2. So the net put out of 2 cycles of pyruvate, because we have 2 pyruvates per 1 glucose, it's going to be 6 CO2, 2 ATP, 2 FADH2, and 8 NADH. And so we're going to look at what that looks like. So we are bringing in pyruvate, we are taking away a hydrogen, we are taking away a carbon dioxide, we're bringing in coenzyme A to make acetyl-CoA. So it will bind with this oxaloacetate so that we can go through the cycle. So we're going to lose 2 carbon dioxides plus 1, that gives me 3. We go through this twice, that gives me 6 carbon dioxides. We made an NADH here, we make 3 here because we're reducing it. That's going to be 4, so we made 8 NADHs after 2 cycles. We made ATP, we did it twice, so we made 2 ATPs, and then we made 1 FADH2. Again, we do it twice because we have 2 pyruvates, therefore we made 2 FADH2s. So again, our net output here. Now, we get to the electron transport chain. So, we're going to have the 2 FADH2 molecules from the Krebs cycle, 10 NADH molecules, 2 from glycolysis, 8 from Krebs. They are now transported to the electron transport chain to produce ATP, because when I think hydrogens, I'm thinking electrons. So they're going to go to the electron transport chain. So the NADH is going to yield 3 ATP per molecule, while FADH is 2 ATP per molecule, which is going to give me the final yield of 36 molecules from 1 molecule of glucose in eukaryotes, and we're going to talk about why. So I made 2 in glycolysis, net-wise. I actually made 4. I used 2, so I got 2 net. Then I made 2 in Krebs cycle, and I made 34 in electron transport chain, so that gives me 38. But then I used 2 ATP from the glycolysis to get my pyruvate into my mitochondria, or to the matrix of the mitochondria, for Krebs cycle. And so I now have 36. The other thing is mitochondrial matrix. That is where my Krebs cycle occurs. Okay? My inner mitochondrial membrane is where my electron transport chain is going to occur, between the inner membrane space and the mitochondrial matrix. At the end of the electron transport chain, oxygen molecules are going to combine with the electrons of hydrogen ions to create water as a waste product. In this case, to remove my hydrogens to keep my concentration gradient. So this keeps going with my ATP synthase pump to make ATP. So in prokaryotes, there's no mitochondria, so we're going to have 38 ATP because we don't bring 2 in. And then because sometimes cells can't use cell respiration, some cells function on short-term and low oxygen levels. Some prokaryotes are anaerobic, meaning they only grow and reproduce without oxygen. So as you see some examples here. And then that's the inside of that one. And so now what I'm going to show you is a picture at the end of this. And so you guys can look at, I'm also going to post this on Schoology as well, the picture of the entire cellular respiration, what goes in, where does it come from. So make sure that you guys look at that as well. And I hope that you guys have a great day, and I will see you this week.